Morning, students. Today's topic is ploidy, chromosomal mutation. So, any alternation in the number or structure of chromosome is called as a chromosomal mutation or chromosomal aberration. So, it may cause changes in the number of chromosomes or changes in the structure of chromosomes. First, changes in the number of chromosomes. Each individual organism possesses a diploid set of chromosomes. So, each organism possesses a diploid set of chromosomes. In human being, there are 23 pairs of chromosomes are present. Among these 23 pairs, 22 pairs are somatic chromosomes and a pair is a sex chromosomes. So the each individual possesses a diploid set of or fixed number of chromosomes. Even though some species of plants and animals having identical number of chromosomes, they will not be similar in characters. So even though some plants and animals possess same number of chromosomes, but their characters is different from each other. So hence the number of chromosomes will not differentiate the number of a species from one another, but the nature of heredity, that is genetic material, DNA, determines the characteristics of the species. So the number of chromosomes does not mention the characteristics of species, whereas the genetic makeup, that is the DNA of organisms, is responsible for the determination of characters. Sometimes the number of chromosomes of spermatic cells are changed due to addition or deletion. Due to addition means a individual chromosomes is added to an existing one, or sometimes if causes a deletion of chromosomes, removal of an individual chromosomes, or basic set from the basic set of chromosomes. This is known as the numerical chromosomal aberration. Instead of 46 chromosomes in human being, either a 45 chromosomes or a 47 chromosomes are present, means it is the numerical chromosomal aberration or ploidy. What is meant by ploidy? Any alternation in the diploid set of chromosomes, either by adding a chromosomes or by eliminating a chromosomes, is called as a chromosomal aberration or ploidy. There are two types of ploidies are there. One is a ploidy involving individual chromosomes which are with a diploid within a diploid set. Individual chromosomes, single chromosomes within a diploid set is called as a aneuploidy. A ploidy involving entire set of chromosomes, that is euploidy. Aneuploidy means ploidy involving individual chromosomes within a diploid set. If diploid set of rise is 24, means if one chromosome is added to the diploid set, 24 plus 1, 25 chromosome, means it is due to aneuploidy. Ploidy involving entire set of chromosomes is called as a euploidy. Suppose the number of a diploid set of chromosomes in rice is 24. If 12 chromosome is added to the diploid set means it is called as a euploidy. Yeah, set is added to the existing diploid set of chromosome is called as a euploidy. Instead of 24, the euploidy organism possesses 24 plus 12, or sometimes both again the diploid set is added. It is called as a euploidy. Aneuploidy. Ploidy is differentiated into two types. One is aneuploidy and euploidy. Aneuploidy is again divided into hyperploidy and hypoploidy. So, hyperploidy is again divided into different type: trisomy, double trisomy, tetrasomy, double tetrasomy, pentasomy. And hypoploidy is differentiated into monosomy, double monosomy, nullisomy, and double nullisomy. So euploidy is divided into haploidy, haploidy, diploidy, polyploidy. Haploidy means monoploidy. So the polyploidy is again divided into autopolyploidy, allopolyploidy. Autopolyploidy is again divided into triploidy, tetraploidy. There are different types of ploidy. Aneuploidy. Aneuploidy is a condition in which a diploid number is altered. That is, diploid number of chromosome is altered either by adding in 21st chromosomes, one chromosome is added, either by adding or deleting. One chromosome is deleted or removed from the diploid set of organism, either by adding or deleting of one or more chromosome in an organism is known as aneuploidy. So aneuploidy is a condition in which from the diploid set of chromosome, that is 46 number of chromosome in human being, from this set, one chromosome is added. Instead of 46, 47 is present, or one chromosome is removed. 46 instead of 46, 45 chromosome is present means such organisms are called as a euploidic organism. So the euploidy are also known as euploids or heteroeuploids. Euploids are heteroeuploids. The euploids are again classified into two types. One is hyperploidy and hypoploidy. The term hyper represent high in number. So the addition of one or more chromosomes to the diploid set is called as a hyperploidy. So the addition of one or more chromosome to a diploid set, that is to the 46 number of chromosome in human being, one chromosome is added or two chromosome is added to means it is called as a hyperploidy. Hyperploidy, the diploid set of chromosomes represent as disomy. Di means two. The two set of chromosomes are present in it, so it is called as disomy. The hyperploidy is again classified into different type, that is trisomy, tetrasomy, pentasomy. Trisomy, tetrasomy, pentasomy. First, trisomy. In trisomy, the addition of single chromosome to a diploid set is called as a simple trisomy. The addition of single chromosome to a diploid set is called as a simple trisomy. So 2n represent diploid set of chromosome. 2n represent diploid set of chromosome. To this diploid set, one chromosome is added is called as a simple trisomy. Trisomy, so addition of to this 21st chromosomes, the addition of one chromosome is added to a diploid 46 chromosomes is called as a trisomy. Trisomy 2n, already the diploid set represented as 2n plus one chromosome is added. It is called as a trisomy. Trisomics is also reported in plants such as Nicotiana, Pisum and Onotria. The addition of sometimes the addition of two individual chromosomes is added from different chromosomal part of a normal diploid set are called as a double trisomy. That is 2n diploid set plus 1 is added. Is called as a double trisomy. Diploid set, one chromosome is added is called as a trisomy 2n plus 1. Double trisomy, two different pairs of chromosomes, one chromosome is added each. That is one chromosome is added in the second diploid set also one chromosome is added. That is called as a double trisomy 2n plus 1 plus 1. Trisomy was first reported by Blaxley in the Thurstromanium Jimson's weed. Later it was reported in tobacco plant, pea plant and Onotria. So in these plants the trisomies are reported. Tetrasomy, addition of a pair or two individual chromosomes are added to a pair or two individual chromosomes is added to the diploid set is called as a tetrasomy. 2n, already it is 2n diploid set, 2n plus 2 chromosomes is added is called as a tetrasomy. Tetrasomy means the addition of a pair or two individual chromosomes are added to a diploid set, that is in eighth chromosomes, a pair of chromosome is added, is called as a tetrasomy, 2n plus 2, and double tetrasomy. If it is 2n plus 2 plus 2, again 2 chromosome is added to the another set of pair of nucleotide is called as a double tetrasomy. Double tetrasomies and tetrasomies are seen in wheat plant. Tetrasomy, this diploid set of chromosomes, a pair of chromosome, to this diploid set of chromosome, a pair of chromosome is added. That is 2 chromosome is added. It is called as a tetrasomic, 2n plus 2 chromosomes. In pentasomic, the addition of 3 individual to this diploid pair, a 3 individual is added to the diploid set of chromosomes. The addition of 3 individual chromosomes from different chromosomal pairs to a normal diploid set is called as a pentasomy. That is 2n plus 1, 2, 3. 3 chromosomes are individual chromosomes are added is called as a pentasomy. 2n plus 3. Next topic is hypoploidy. Hypoploidy means loss of one or more chromosome from a diploid set is called as a hypoploidy. That is, it is a diploid set of chromosome, a normal chromosome. From this normal chromosome, a removal of one or two chromosomes takes place is called as a hypoploidy. The hypoploidy is again classified into two types. One is a monosomy and nullisomy. Monosomy, a loss of, it is a diploid set. From this diploid set, a loss of one chromosome is called as a monosomy. From a diploid set of chromosome, a loss of one chromosome is called as a monosomy. Monosomy is represented by 2n. 2n means diploid. From this 2n, one chromosome is removed. So if in human being chromosome number is 46, due to monosomy, the 45 chromosomes are present. However, the loss of two chromosomes, 3 individual chromosomes are called as a double monosomy or triple monosomy. So, double monosomy or triple monosomy, a loss of 2 individual chromosomes from a different set of diploid chromosomes, or 3 individuals are removed, is called as a double monosomy or triple monosomy. Double monosomy is represented by 2n minus 1 minus 1. Triple monosomy is represented by 2n diploid set minus 1 and minus 1 is called as a triple monosomy. The double monosomies are observed in maize plant. The double monosomy is observed in maize plant. The next, nullisomy. Nullisomy, a loss of a pair of homologous chromosomes is lost from the diploid set is called as a nullisomy. A homologous chromosome, a loss of homologous chromosome from a diploid. It is a normal diploid chromosome. From this normal diploid chromosome, a homologous pair is removed is called as a nullisomy. Nullisomy is represented by 2n minus 2. And double nullisomy is also observed. 2n minus 2 then minus 2 is called as a double nullisomy. What is meant by nullisomy? A loss of homologous pair. A loss of homologous pair from a diploid set of chromosome is called as a nullisomy. Nullisomy is represented by 2n minus 2. Sometimes 2 homologous chromosomes are lost from a diploid set is called as a double nullisomy. Double nullisomy is represented by 2n minus 2 then minus 2. Selfing of monosomic plant produces nullisomics. Selfing of monosomic plant produces nullisomics. They are usually lethal. Lethal means it causes death of the individual. So, if a pair of homologous chromosome is removed from a diploid set, it causes the lethality to the organism. Euploidy. Euploidy is a condition where the organism possesses one or more basic set of chromosomes. One or more basic set of chromosomes is called as a euploidy. Euploidy is again classified into monoploidy, polyploidy, diploidy, and polyploidy. So the condition where the organism possess stomach cell has two set of chromosomes. All the organisms are made up of a chromosome in paired nature. Two set of chromosomes are present in the organisms. So it is called as a diploid. That is diploid. If the organism possesses two set of chromosome is called as a diploid chromosome. The organism possesses half a number of chromosomes in a somatic cell is called as a referred as a genetic chromosomes are called as haploid. Only from a diploid set, one chromosome is present in each set of a chromosome is called each set is called as a haploid. That is monoploid, haploid chromosome. It should be noted and haploid is represented by N. Diff is N. Haploid set is represented by N. For example, the common wheat plant is a polyploid. Polyploid means it possesses more number of chromosomes, either 3 chromosomes in each set or 4 chromosomes in each set is called as a polyploidy. So the number of chromosomes in wheat. For example, the common wheat plant is a polyploid, that is hexaploid. The diploid nature is 2n equal to 6x hexaploid, 72 chromosomes. It is haploid number is 36, but it is a monoploid. 36 is a monoploid, but as X is 12, where X is monoploid, X is a 12, haploid is a 2n, that 2n is a diploid 72, its haploid is a 36, the monoploid is 6x is 12, X is equal to 12. The haploid and the diploid condition came regular after another, and the same number of chromosomes is maintained from generation to generation. So that is the diploid. For example, if the human being diploid number is 46, from this haploid is a 23. It is the number of chromosomes, whereas monoploidy occurs when an organism is under polyploidy condition. In a diploid, both the monoploid and haploid chromosome number are the same. If it is diploid means the individual, the both monoploidy and haploid chromosomes are same. When it is polyploidy means the number of haploid and also monoploid is varied. Thus, a monoploid can be a haploid, but all the haploids cannot be a monoploid. That is, all the haploids cannot be a monoploid. The next topic is polyploidy. Polyploidy is the condition where the chromosome possesses more than two basic set of chromosomes. That is, more number of chromosomes are occur is called as a polyploidy. Polyploidy is a condition in which more number of or more basic set of chromosomes were occur. Where there are three. It is a haploidy, diploid, two. A set of each chromosomes are in a set. If it is polyploidy means a 3, that is triploidy, or 4 means tetraploidy, or 6 numbers means hexaploidy. These conditions are called as a polyploidy. If it is triploidy or tetraploidy or hexaploid are considered to be a polyploidy. Generally, polyploidy is very common in plants, but they are rare in animals. An increase in the number of chromosome set has been the important factor in the origin of a new plant. A new plant species arises due to alternation in the change of chromosomes. But higher ploidy level leads to death. If it is more ploidy means that is any alternation in the chromosome means it causes death. Polyploidy are of two types. One is autopolyploidy and allopolyploidy. First, autopolyploidy, the organism which possesses the more than two haploid set of chromosomes. If the number of chromosomes is more than two haploid means it is they are derived from within the same species is called as a autopolyploidy. So which is derived on the same species, only single individual, the number of a chromosome is increased, is called as a autopolyploidy. It is divided into two types. One is autotriploids and autotetraploids. Autotriploids, three set of its own genome. Genome means the totality of the DNA is in the form of three sets. Three sets occur. They are produced artificially by cross between the autotetraploids and the diploid species. That is, autotriploids are caused by artificially crossing between the autotetraploid. Tetraploid means four set of chromosomes and diploid means only two set of chromosomes are crossed. They produce as autotriploidy. They are highly sterile. These plants are highly sterile. Sterile means they are unable to reproduce next generation. So it is caused due to defective gametes formation. For example, the cultivated varieties of bananas are usually triploids and are seedless. The cultivated bananas does not contain seeds. Only the wild variety of banana contains the seeds. So they cannot be able to reproduce to the next generation, having larger fruits than the diploids. So the fruit size is also varies in the diploid banana to the triploid bananas. The triploid bananas does not have a seeds, and the size of the fruit is also larger compared to the normal banana. Triploid sugar beets have higher sugar content than the diploid. Similarly, sugar beets are having a higher sugar content than the diploids. The sugar beets, that is triploid sugar beets, also resistant to the molds. Molds are fungi. They are highly resistant to the fungi. The next common dupe grass, in Tamil it is called arugambil. Binomial is Cynodon dactylon. As the natural autotriploids, the natural autotriploids. They are naturally produces triple number of chromosomes without any crossing. Seedless watermelon, apple, sugar beet, tomato, bananas are some of the example for autotriploidy. The next, autotetraploid. Autotetraploids are the individuals which are having a 4 copies of its own genome. The genomes are in a 4 copies. They may be induced by doubling the chromosomes of diploid species. Example, rye. Rye means Tamil it is called as kambu, grapes, alfalfa, groundnut, potato and coffee are some example for the autotetraploid. Triploidy individuals, the chromosomes numbers are 3. Triploidy, the chromosome numbers are 3 in each set. Tetraploidy, the number of chromosomes are 4 in each set. That's autotetraploidy. Allopolyploidy. Allopolyploidy and an organism which possesses 2 or more basic set of chromosome. Organism which possesses 2 or more basic set of chromosome derived from 2 different species is called as a allopolyploidy. Allopolyploidy means 2 or more basic set of chromosomes derived from 2 different species is called as a allopolyploidy. It can be developed by interspecific crosses, and fertility is restored by chromosomal doubling with colchicine treatment. Fertility means the fertile factor of the individual can be restored, retained by means of the chromosomes doubling, which takes place with the treatment of a colchicine. The function of the colchicine is to double the number of chromosomes. If it is sterile, means the sterile plant can be making into fertile by means of doubling the chromosomes. Allopolyploidy are formed between 2 closely related species. For example, the allopolyploidy plant is Raphino brassica. The Raphina brassica is a allopolyploidy plant. Raphina brassica was first introduced by the Russian scientist Karpychko. He developed Raphino brassica. It is a cross variety between the Raphinus sativus Aphanous sativus is a radish. It is a seeds of radish and cabbage, Brassica oleracea. Its chromosomes number is diploid as 18. Both the plants are having same number of diploid set. 21, 2n equal to 18, 2n equal to 18. To produce F1 hybrid, that is young ones, which was sterile. The F1 hybrids are sterile in nature. That is sterile means it is unable to reproduce next generation. When this sterile plant is again treated with the colchicine, the chromosome number is doubled. 2n equal to 18, it is a sterile plant. The number of this chromosome is doubled by means of a colchicine treatment. It becomes a tetraploid. That is 2n equal to 4x equal to 36. The number of chromosome is 36 in nature, in gap. So the scientists expected that the plant possess the roots of radish and the leaves of cabbage. So the roots like the radish and the leaves like the cabbage, which could be made the entire plant edible. Both the root part and the leaf parts are edible. But in this case, it was a great disappointment. That is, the root shows the cabbage character and the leaf shows the radish characters. The root shows the cabbage root and the leaves are like a radish. So this the Rabido brassica is a failed variety. The next example for autotetraploidy is triticale. The triticale is the successful first man-made cereal. The triticale is the first man-made cereal which is crossed by means of two varieties, that is triticum durum wheat and secale cereal rye kambu. A cross is made between a wheat and the rye. It produces the triticale plant, that is tetraploid cross between a diploid wheat, diploid 2n equal to 28, diploid wheat and a diploid rye variety 2n equal to 14. Here the chromosome number is changed. Wheat 2n diploid set is 28, in rye diploid set is 14. Hexaploid, the cross between the tetraploid wheat T4x tetraploid represent 4x tetraploid, here diploid 4x tetraploid wheat triticum durum is macaroni wheat. It is commonly called as macaroni wheat, and here rye, diploid rye is crossed. In the F1 generation, it get a triploid 21, that is triticale, which is a sterile plant. The sterile plant can be treated with a colchicine treatment. Chromosome is doubled to become a hexaploid. So a cross between a hexaploid wheat triticum aestivum, it is a bread wheat, and a rye. The hexaploid triticale hybrid plant demonstrate the characteristics of both the macaroni wheat and also the rye variety, that they combine the high protein content of wheat and the rice high content of the amino acid lysine, which is low in the wheat. The amino acid lysine is low in the wheat. It can be increased by means of crossing the wheat with the rye. The hexaploid triticale hybrid plant demonstrate both the characteristics of macaroni wheat and the rye. So this possesses the macaroni wheat habit and also the rye one. So the macaroni wheat contains high protein. The macaroni wheat contains a high protein content and the rye contains the lysine amino acid. Both characteristics are found in the hexaploid triticale, which is having the high protein content and also amino acid contains, which is absent in the amino acid is absent in the macaroni wheat. But the triticale possesses both amino acid lysine and also wheat.